Hi, Year 12. Sorry about this. I lost my first one. So, topic 5, key assessment 1. Okay. Now, because I lost it, I've already done some annotation here, but I'll take you through that. The pigment content of mountain plants can be affected by various environmental factors. These include altitude, UV light, and temperature. The pigments, chlorophyll found in chloroplasts and flavonoids found in sap vacuole. Flavonoids can protect plants from UV radiation, which can damage DNA. Now, as soon as I've seen damaged DNA, I've written mutation. State the location of the chlorophyll in the chloroplast. This comes from the light-dependent reaction. The chlorophyll is excited and the electrons move down the electron transport chain. We know that that's in the thylakoid membrane. Okay. The altitude in which the plant grows affects the flavonoid content. Devise a procedure to show the increase in altitude on one species of plant growing on the mountain. Now, what I'd said earlier before it got cut off is when you see devise, you're automatically thinking that, okay? And you're given some of this in the question as well, all right? So our independent is altitude. How are we going to measure that? Our dependent is the flav content. How are we going to measure that? Then we're going to have some control variables, but not that one, okay? Because we're given that one, all right? We're going to talk about repeats. And what CPACs are we talking here, okay? Now, we'll talk about that as we go through. Now, the first thing is the IV. We need to have a range of about 5, okay? 5 different heights or altitudes. Now, you'd have seen this sampling in the live lesson, okay? So we know that horizontal distance is not affecting us when we're looking at altitude. So you've got to look at altitude here. All right. We're going to measure the flavonoid contents. All right. That's not something we can do in the field because back here it tells us that the flavonoids are in the sap vacuoles. So we're going to have to somehow get that out of there. So in terms of the DV, we need to actually collect some of those plants, okay, or grow some of those plants. In terms of control variables, well, let's think about how we're going to standardise the plant material itself, mass, okay, the part of the plant maybe, all right? And for a second control variable, let's think about all those abiotic factors which we've been learning about, all right? We know that we're moving altitude, so we can't really control temperature, all right? And I would argue that some of these other things we're going to struggle to control. But if you look, there's all sorts of other abiotic factors you can take into account there. Humidity, soil pH, wind speed, okay? Those bits are a bit more straightforward, but then we need to actually work out what the CPAC is about. Now, this is all sampling. So this is CPAC 10, which you haven't done, but which was covered in that live Field Studies Council lesson you saw on YouTube. But this is where it's difficult, this question, I think, because we need to measure the flavonoid content. We're told that that is in the cell sap vacuoles. So you need to think about an experiment that you've done where you've removed pigment from a cell sap, okay, from a vacuole. Now you've done that in CPAC number 3, all right? So how did you do that? We need a way of extracting it, okay? Best thing here really is solvent, something like alcohol, all right? I think you probably would be allowed something like high temperature here, okay? And then how are you going to measure it? Well, if you think about that CPAC number 3, that beetroot experiment, we measured it with a colorimeter, okay? This is a good example of a question where you're going to get so far with your standard IV, DV, CVs, but you're really only going to get right to the crux of it by actually working out what CPAC it is talking about. Not easy. Right, this is a data question, okay? So, scientists investigate the effect of UVB on both chlorophyll and flavonoid. A group of plants were exposed to UVB for 20 minutes per day for 1 month. The chlorophyll flavonoid content of each plant were determined. This was repeated for 3 more groups of plants. Each plant was exposed for different lengths of time. All variables are kept constant, so we've got valid experiments. Okay, compare and contrast. Compare similarities, contrast differences. The effect of length of exposure to UVB on the chlorophyll and flavonoid content of these plants. Okay, so let's look at the similarities first. Okay, well, if you look at chlorophyll, it's just down the whole time. Okay, if you look at flavonoid and this is a good way of doing this, drawing this on the thing. It goes down, all right, and then it comes up, if that makes sense. So from 24, 30, so it goes up, okay? So the similarity here is that for 20 minutes, they go down, okay? And then obviously the difference is that once we go past 20 minutes, all right, chlorophyll goes down, flav goes up, all right? And then if you look at the third marking point, well, here for flavonoid, you're going from 25 to 40. So that's a change of plus 15 overall. And in this one, you're going from 32 to 24. So the change there is, how's my maths, minus 8. So the overall change in flavonoid, okay, is greater. So always look at that, try and manipulate some data, okay, when you're looking at these sort of data type questions. That's where you're getting that last mark. And then this last 5 mark, which is tricky as well. As altitude increases on the mountain, temperature decreases and the UVB radiation increases. Altitude, temp, radiation. Scientists have found that the rise in temperature decreases the flavonoid gene expression in plants. So you're told this, okay? Analyse the data to discuss why altitude can affect plant growth. Plant growth is vague, okay? So what does that mean? What causes it? So plant growth is going to be about photosynthesis. Yes, that's what's causing plants to grow effectively. I've circled altitude because everything we say in this question has got to be in respect to altitude, starting it off, okay? So if we look at the data simply, as altitude increases, mountain temperature decreases and UV radiation increases. So as altitude increases, flavonoid increases. Okay, because why? We go right back to our question here. Flavonoids protect from UVB. Okay, so the more altitude we get, the more UVB we get, okay, and therefore the more flavonoid we need. Okay, why? And this is where my dots come in, my therefores, okay? You're going to protect your DNA. Or something like less mutation. Oh, sorry, that's wrong, that's the next bit. So you're protecting your DNA from UVB, so some acknowledgement to UVB, and therefore less mutation, okay? All right, and if you get mutation, you might have an effect on growth. Okay, so there's the first bit. What else do we know in this question? Well, we've got to go back to this table, okay? Because we know that actually, as we increase the UVB, i.e. we increase the altitude, we also start to reduce our chlorophyll, okay? And that's where this growth comes in. So we've got less chlorophyll, therefore, okay, we've got less photosynthesis, okay, and therefore less growth, which you're not going to get because you've got growth in the question, all right? And then the last marking point that they're accepting here is just the idea, because they're talking about temperature decreasing, you can manipulate that, and they're talking about less enzyme activity, and therefore less growth. All right, so one to sort of pick the bones out of and think about how you're going to approach that question. Lots of good highlighting here in the question, if I do say so myself. All right, I'm focusing my attention on altitude. All right, I've said something about temperature. Okay, and then I'm looking at, right, plant growth. So I've gone, okay, plant growth to do with photosynthesis. What's happening there? And altitude. All right, I've looked at flavonoids over here, okay, against UVB. All right, but I've also looked at against information we're getting in the question here. Okay, so that's how I would go about answering that question. As I said, it's been a tough week, I think, learning this photosynthesis stuff and things like that. Not the easiest thing to learn remotely, okay, but hopefully you've given it a good go. And actually that question doesn't have a huge amount about photosynthesis in it. There's quite a lot of synoptic knowledge in there, okay, so testing some of your previous work. Okay, stick with it. As always, if you have any questions, feel free to email me and I'll be at the other end. And I hope you're keeping safe and well, and look forward to seeing you all soon. Take care. Bye-bye.